Hello, my name is Christoph Kutscher. I'm very happy to have you. I'm the founder of Farm to Farm, and with Farm to Farm we produce videos about sustainable agricultural systems, especially arable farming. And we did a lot of videos and a lot of expert interviews. And one of these interviews was a webinar with Dr. Alberto Andrino from the University of Hannover with the topic of the Arbuscular mycorrhiza. And this webinar was live in English. He held his webinar in English. I translated the presentation, the general part of the webinar, in German and posted it a couple of weeks ago. And now in this video, I want to upload or show you the questions and answer part of Alberto, where he answered the questions from my side and from the participants of the webinar. And it is very interesting, and I think it's the best way and the most authentic way to keep this part in English. I hope this is fine for you. Usually all my videos are done in German, but anyway, let's do it. Let's try it this time. Let's do this, and let's go into the question and answer parts of the webinar from Farm to Farm with Dr. Alberto Andrino. Can I start with some questions I wrote down?
Yes, please.
Speaking about the AMF itself, or how it works, I was always wondering, or not. Let's put it different. I heard that the fungi grows faster than the plant's roots. So, is there any scientific proof of that? That if the fungi itself, the hyphus, grow faster than the roots of the plants as soon as they are connected?
I mean, this depends very much on, of course, something I probably didn't comment in detail in these challenges. I was not talking. This depends very much on the partnership they establish. Of course, there are better host plants that do establish like a better symbiosis with a better performance, meaning that there is a better translocation of carbohydrates or lipids. And this probably affects the way the hyphae can grow and the fungi can develop. But I wouldn't generalize that there is the fungi, I would say the growth, I would need to probably check this, but I think the growth of the hyphae itself is not that fast. I think root hairs are really, really fast. Within hours, you can have, compared, for instance, to saprophytic fungi, which are growing within hours. The growth of arbuscular mycorrhizal fungi is a little bit slower. I would say it's a little, but it's very dependent on the host plant and, of course, the soil conditions. So I think here, I would say there's not a general yes or a general no. I think there are several gray scale here, because I don't see. Yeah, it depends very much on the host plant, I would say here.
Okay, and if I can go on, there's always we have to, or AMF. First thing, we're speaking about AMF, and there are a couple of different species of AMF. But what is if we speak 80% of the plants are working together with AMFs, are they working with all the different species, and how many are already done or researched a lot?
Look, morphologically speaking, there have been identified around 200 no more morphological species. So the amount of species represented in the Glomeromycota division are rather low. The thing is that they are the same species you isolate in an Mycorrhizal. You can, like Rhizophagous irregularis, then you go to another soil and they perform completely differently. So morphologically they are the same, but they do behave differently. Their capability, their abilities to mobilize nutrients is completely different. So there, and also you may have in one, because most of the research done to talk about the ability of mycorrhiza are done on controlled experiments. And there you may play with 1, 2 species at the same time, 3 species in the best case scenario. But in nature, you may end up having the same plant, talking about a tree, establishing mycorrhiza with 5, 10, 15 different species, but not only arbuscular, also ectomycorrhiza, like poplar. Poplar, for instance, has a first stage of arbuscular mycorrhiza, and then when it's becoming a little more mature, it turns into ectomycorrhiza, and at some point they could share both type of mycorrhiza and different species at the same time. So yeah, and I think this 80%, it's like a repeated number you can read in all reviews. And it would be great to go to the source, no? Because I read also 75, 80. So this was a number I took. But what I want to say is that with this number is that it's widespread all over the terrestrial ecosystems. And more importantly, that they co-evolved with the plants. So plants were able to conquer the land together with the, and I think I have one more slide here. No. Are you seeing now the presentation?
Yes.
This is not something. Yeah. So like 450 years ago, 450 million years ago, the first organisms that were looking like arbuscular mycorrhiza were identified. And since then, the plants have co-evolved together. That's why this widespread is so common that first they started the arbuscular mycorrhiza, and then 100 million years later, the real Aryan mycorrhizas, but the first prototype of arbuscular mycorrhiza was already found in 460 in some fossils. I need to check these things. Yeah, so yeah, they are widespread, and
because they have been together since ages.
Yeah.
Besides this part of Farm to Farm, we also offer a couple of products as a company. We're a young company and we focus on sustainable products. And one of these products is Mycorrhizal inoculant, a very highly concentrated inoculant, which you put on the seeds as a seed coating, and it is used in order to get a very well establishment of the mycorrhiza in the soil for your plants. If you're interested in this mycorrhiza inoculant, come to our website farm2farm.eu. Beside this product, we also offer, and which is also a very important part of our company, is cover crops. We produce and develop multi-species cover crops. If you are interested in this, write me an email or write us a message or come to the website farm2farm.eu. I would be happy to have you. Okay, Thomas is asking if he understands it correctly that that plants who need more phosphorus or have a higher need of phosphorus are working together better with AMF. Or is this connection not right? So if
we are talking about natural ecosystems, yes. A plant that relies its phosphorus acquisition on AMF are succeeding better than plants that do not hold an arbuscular mycorrhiza. But when we talk about agricultural ecosystems, this changes. As I said, because we were since agriculture started, we've been selecting species that respond better to fertilization. We were trying first with manure, later on with first with guano, manure, any kind of fertilizer, and therefore we were selecting species that were working better with inorganic phosphorus in the form of all these fertilizers, better than with the AM fungi. So I don't know if I'm responding to his question.
Yes, I think so. I mean, I would what I can add with my very basic knowledge that is that there is also knowledge about that. For speaking about the agricultural plants we use in Germany and Austria, there are differences between the crops, and as far as I know, for example, corn is very good responding to AMF in the soil, whereas cereals are not that much responding.
Exactly. Yeah.
Okay.
Yeah, cereals in general are not responding, or probably, but this is again, you can when you search in the bibliography, probably that's what I mean in the last challenge. So we probably don't need only to focus on the yield, because yield, of course, in the end, we need to evaluate everything on the yield and on the prices we get from our production. But thinking on the long-term effects, this stabilization and somehow water retention in the long term are also benefits we need to consider. And these benefits are present also with the cereals. So
yeah, sure. I hope we answered the question from Thomas. If I can go on, I would like to ask if there is knowledge about the AMF and the different pH of soils, because usually if we speak about fungi and pH, there are certain pH values which are better, or where fungi feel more comfortable. The question is what about AMF? If AMF is all over the world, I would assume that it's not that sensitive to pH. So my question is about that.
Look, you were talking about, look, you can, I will share my screen now, and let's see if I can you see, no, you still see the presentation, no?
I see the presentation, yes.
Okay, let me change the screen because I want to share with you, there is a database that, I simply quit the presentation, and I go here. Are you seeing this from this Marjam? Can you see?
Yes.
Okay, here you can play a little bit around, because this database is created by Marja Opik. She's doing quite nice things with arbuscular mycorrhizal fungi, because she's searching. She's somehow by location, by biome, and if you click here, you can search if this fungi was isolated in a culture or in a grassland. And at some point, I know she's updating, and her group is updating also the soil conditions, which are also connected to the pH. And if you have, I can send this link to you. I found it very interesting, especially because she's quite good at developing tools, molecular tools, to describe populations in soil, because the problem you have when you are working with arbuscular mycorrhiza is to find a good group of primers that allow you to describe the populations you have in soils, and to determine whether you have a more or less rich soil with arbuscular mycorrhizal fungi. And here, and there is a paper also associated to this database. Here you have, I think there is something about the biome, the location, grassland culture, anthropogenic ecosystem, and here you have temperate agriculture, so abandoned field, and so you can come here and you will find common. It's amazing what they do.
Yes.
So if you have any interest, you can go to here to experimental plots, anything, and then you can see the list of fungi they are finding, and then you have tools you can utilize to make your own groups, to find different so in the mice field, which are the species you may find. So
Okay, interesting. So there's another question from Clemens. He's asking a very interesting question, and a very often occurring question is, how can you count, or how can you understand how much AMF is in the soil, on the field? You know, as a farmer, I would like to know how much AMF do I have, and second question is, what shall I do to improve the DMF in the soil?
I think I'm searching for. I like this this website a lot. Probably you have seen it or not, but here
No,
you
no,
can, yeah, this one is great. You come here and you have methods, and then you come to Arbuscular mycorrhiza, and here you have all the standardized methods to go to the field, take your plant, sample your plant, the roots, stain the roots, as I was showing you these pictures, and you can count colonization by length, by root length. This would be one way, okay?
with the microscope.
With the microscope.
But is it a way that a farmer can do with a microscope? Is it?
Yeah, if you want, we can even prepare a session where we, with very little amount of money, I mean, with vinegar and very cheap things, and nowadays you can buy a very easy microscope for little money. Even these ones which are connected to the cell phone, you can check this. And this could be one way, of course, checking the roots. This is one way, and the other way is also to do the I mean, this is the most used way, but also if you want to see if your soil is containing more or less spores, there is a way. I would need to check this, but you can also extract the spores from your soil. So imagine you don't know if your soil contains any arbuscular mycorrhizal fungi. There is a way that you can use some sieves with different sizes, and you can just filter your soil, and then in the end, also with the microscope, check if you have some spores. What is this? But this website, it's really nice. It's and then you would be looking at spores, and also you can do your own isolates in your farm. So in your soil, you take some soil, isolate the spores, and then there are ways to do what is so-called a trap plant. You take your spores with some sorghum, which is a perfect trap plant. A trap plant simply, it's a plant that establishes mycorrhization very easily, produces a lot of roots, and all the roots are full of spores, hyphae, and then you simply use this as inocula, like probably the inocula, I don't know, for instance, Rutella or the inocula.
They use also sorghum to
Exactly. And this is the way you can do it. Not all the species are good to develop and grow with sorghum, because like, for instance, like Aceulospora, some general like Aceulospora, Scutellospora are difficult to grow in trap plant systems, but yeah.
But is there also a possibility to, like as a farmer, you are used to take a soil sample, send it to a lab, and get the results for chemical analysis? Is this possible also for AMF?
Yeah, I would say the easiest and the fastest way is to, as I said, it could be to take a core of soil core or a root sample, just 15 centimeter, 20 centimeter. If it's maize, just separate like 20, 30 centimeters from the shoot, and then dig a little bit, take some roots. And the procedure is rather easy. You need not very expensive material, and if you want, we can probably talk. Probably we could do something like a practical lesson and show how to do it, because this is very informative. Because every time you apply Arbuscular mycorrhiza, also you can test whether your plants are getting this mycorrhiza. This is first thing, and also if you have an area like a fallow area where you applied last year, but this year you are not planting, you leave it 1 year and you don't apply any other Arbuscular mycorrhiza, and then you plant again, it's very likely that some spores are still there, and you will again get your plants mycorrhized. Yeah.
The question is
to count for this, no?
To count and to know how much is how much, like if you know what I mean, you need a feeling, if is that a lot or is it not a lot what I see?
Yeah. There are several ways. I mean, also the roots change a little bit. Its color, they turn a little bit yellowish, because once the fungi is inside, also they also turn to change a little bit the color of the root. But it depends very much on the plant species. but I can prepare 2-3 slides and send them to you with
Yeah,
the
would be very interested.
methods, at least with a list of methods. I mean, this webpage is great, but then you have the inbound here. This is another web page. I would say it's a reference, because you come here to methods, and you can quality control, and infectivity assays. So here, all these methods are available. You can read them. You can determine the mycorrhization, how to do it. And I could help you with this. So it is.
Yeah, it's interesting.
Yeah.
Cool.
And it's the only way, I would say, nowadays. I mean, of course, you can also have some indirect proxies, like checking the photosynthesis, but you don't need to buy the device itself. You probably know this SPAT. That in maize is very used. I know, I was working with researcher. So it's like a device, you clamp the leaf and it measures how green the leaf is. And if you have different treatments with arbuscular mycorrhizal fungi without, normally this SPAT measurement is also giving you the idea that plants are doing better photosynthesis. This is an indirect way, of course. Yeah, biomass is the most used one. So comparing always, I mean, always you have to compare to something, no? But for the farmer who is just trying to identify if the plants are getting the mycorrhiza and are getting the benefit, you need to look at the roots. This could be the only way, I would say. And really, it's not difficult. It's difficult probably to get a good staining, but once you've seen the root, mycorrhized root, you never forget how it looks like. And someone who is just doing it once or twice a year, he or she will learn it very easily.
Okay, cool. One more question from Clemens is, what about the tillage? What influence does have tillage to the colonization and TMAF?
Yeah, in theory, it's the worst practice you can do, together with fungicides and excess of P. But again, this depends very much on the type of tillage, or even if you do, in theory, this direct seeding with mulching could be the best practice you can do. So zero tillage, and you keep your dead plants over the year. You just somehow compress them a bit and then direct planting. This could be the best practice, because then you are sure all the mycelial network is still established. But here, so the bibliography here is quite clear. What you may find is a reduction of the diversity. So if you are plowing or you are tilling the soil, you may find a reduction in the species of arbuscular species. But this could be one of the downsides of tillage. Of course, you break physically the connection between plants and the fungi. Even when you collect the plant, you still let there some propagules, so dead roots, which are containing the spores, and the spores themselves are there. Therefore, if you are doing tillage, you normally also kill the fungi, because you break the connection to the roots that was already there. So yeah, that's also part of the management. I was also proposing in these 4 challenge points in the challenges, no? That we try to find the threshold of management, or a certain management where, yeah, AMF are developing in a good way, and we still keep the benefits. And also, I mean, if we reduce tillage, we also reduce consumption of fuel and everything. So I think everything goes always into the same direction: reduce resources.
Yeah, and so the answer is the less tillage, the better for the AMF.
Yeah.
And you spoke about that if you have this mycel in the soil, and if you don't destroy it by tillage. My question is, how long, if you harvest, let's say you harvest corn and you have a lot of mycel, and then you leave the soil from October until May next year bare as a fallow without plants, so the AMF itself cannot grow or live anymore because it doesn't get any carbon. So the question is, how long does this mycel keeps not alive but keeps active or is potentially active under such conditions?
Yeah, this is a very good question, and I don't have a clear answer for that, because it depends very much on the soil. It depends very much on the water retention capacity of each soil, and depends very much because in the soil, all the processes, all the metabolism you can think of, it's always very slow, and especially during winter, even the microbes are also developing much slower. The metabolism is also slowed down, and the good thing of all these AMF, most of them produce thousands and millions of spores. So they always let a legacy to go on with their infection year by year and season by season. So there will be always a chance that there's some living spore. It's always a matter of probability that the root finds this spore. There is signaling. The signaling.
Can you get deeper here? How this works? Because the soil is huge. So and there are a couple of spores and there's some root. How does it come together?
This is a complete chapter. So there is so indeed the fungus cheats the root to promote the infection. So because it behaves like a parasite, so it has to break the cell wall of the root to enter the root. And the fungi already develop all this cheat signaling to tell the plant, look, I'm not dangerous, let me enter, and then establish. But there is a lot of still ongoing research on this mechanism. And your question probably goes more into how, so I don't know, it's more how is it happening, or why, or if it happens, at which level it happens, depending, I think, or sorry, may I?
No, go on.
I think first of all, there is a probability. No, there is the probability that the root and the fungi find each other. That's why when you apply this inocula, you are, for instance, this Rutella, you are basically increasing the probability that the mycorrhization takes place. But it could also happen that the fungi you are putting in the form of inocula, it can be also displaced. So it first infects the plant, but since probably it could happen in some soils, that's why probably it's something you could tell me. The same inocula is not working the same in every soil. Probably you've heard in some farmers telling you, this year it worked pretty well. Or the same here in this soil, it's doing 20% better, I get more production, and the other one I didn't find anything. I don't know, did you get any information like that, or it's always quite constant? Is it
of course, there are huge, depending. But what I always try to say is that the
trend.
Yes, the trend. Of the one hand, on the other side, it's a complex system, and let's start it. It's always a trade. So if as far as I understand, if the plants is in a comfortable situation with nutrients and water and everything, it is not that much interested in investing in the mycorrhiza. So if you have a control, if you have a trial and with a control, and both plants are under very good conditions, you won't have that big differences, because the plant is under best conditions anyway if there's a lot of nutrients and water. So what we see, and then what groundwork is showing us on results, is that the worse the situation is, the more stress the plants do have, the better the results are with inoculation. And so I hope that covers also what you see in research.
Yeah, exactly, because the same various species, even during the year, can also have different performance. And that's also what I wanted to say. So in the end, what you do when you inoculate the soil, you are basically increasing the probability that the spore and the root are going to find each other, and they are going to establish this beneficial symbiosis. I mean, nowadays, there is also a lot of research, especially on cereals, that they want to encapsulate the seeds with the spores, so that they want to have this typical encapsulation of seeds where you put sometimes fungicide or nutrients just to promote the, but nowadays there are several tests. During the last 10, 15 years, they have done a lot of trials to include also spores, so that when you are seeding your the field, you already get mycorrhizal plants since the very, very beginning. So, because the idea is that you want to increase the probability that the spore and the plant finds each other. So once they find each other, the signaling occurs within micrometers, and yeah.
They have to find each other, and or just to understand it, what's the distance they can speak to each other? Is there any? That's what I'm.
I would say it's rather small, because I mean, but you have to think also that when the roots are growing, all the exudates are not only activating this fungi, are activating many other microbes, and there is still a lot of signaling we are not able to understand, and we didn't. There is a lot of research on this, but yeah, the distance, I would need to read about that. I don't know. Actually, I don't know. I would say it's rather small, I would say. But it also depends if there is a pre-established mycelial network. By avoiding tillage or by having some other management, you will always have a better chance to have a faster and more efficient colonization of your roots.
Okay, so there's one more question about what if the soil has too much soluble phosphorus in it? Is that bad for AMF? Or for the symbiosis?
Yeah, in principle, yes. I mean, unless you may find or select one species that it's adapted to agricultural management, where you have a lot of P input. Normally, high content of phosphorus in the form of fertilizer is not good for the fungi, because I mean, once the plant can take up enough phosphorus with using the direct pathway, as I explained in this slide, there's no any need to establish, and this can be easily seen in many research publications. You just play a little bit around with the P. You start with the highest, just the typical Hoagland solution is super rich in phosphorus, and you just start decreasing. And I would need to check, but I think it's something like 3 micromolar phosphorus. Then it's really, really low, and there the fungi really boosts the growth of the plant, because it's not enough for the plant to take it up over the direct pathway, and there the fungi really operates and gives the best performance to the plant.
Okay, and so there is we cannot really. You can probably not really answer the question: how much shall you reduce phosphorus fertilization if you want to improve the AMF? So, do you know what the question is? What the question is? You know, like if you're a farmer, you're having your recommendations of fertilizing phosphorus in order to feed the soil and feed the plants. And the question is, how much shall we reduce it as a farmer in order to keep the system of AMF forward?
Look, the thing is that arbuscular mycorrhizal fungi do not only mobilize inorganic phosphorus, they can also access organic, like I said, phytic acid, for instance, or this phosphorus, which is forming part of the dead plant material. Sorry. Therefore, it's every decision made to reduce phosphorus in the form of fertilizer should account not only for the easily available phosphorus present in the soil, also in the organic forms, because there is also part of this phosphorus can be accessed by the fungi. meaning that every phosphorus in the form of primary minerals, organic phosphorus, and also the phosphorus in soil solution coming from fertilizer can be accessed by the fungi. That's why it's difficult, as you are saying, no? Like there's no any universal at the moment. There's not a universal recipe that you can tell. Look, you reduce 1/3 now in autumn, and then you just do 1/5 or 1 to 2 in summer. This has to be tested, and that's why I think the intensive agriculture is not still using intensively AMF, because you need this previous screening of the plant species that do develop well with under low P conditions, and still the yield is not affected, but it still responds good to the mycorrhization. Yeah, sorry, I cannot tell you. You need that's 20%, but you can play a little bit around with this. I mean, there are, when you ask for the soil analysis, you get this available P, no, and you normally calculate how much you need to put in plus, like for in the form of fertilizer. And this could be probably done in 2 or 3 levels, just as a rule of thumb, like reduce 20% at the beginning and see what happens. But this is probably not something the farmer want to listen. So they want to listen, no, you told me this, and now my plants are small and I don't have production. That's why, as one of the challenges I was also proposing, the breeding of the new species have to be done together with the fungi. So companies like KWS or seed companies like DuPont or Syngenta, they would need to start working hand by hand with researchers working on symbiotic organisms, because then you would be selecting the best breeding, the best plant species, which on the one hand establish good symbiosis and also are somehow adapted to variable amount of phosphorus.
I mean, that's something that that is not only with AMFs the issue, but it's with all the biology in the soil that farmers need to understand that it is a little bit like a weather forecast. As the farther you want to predict what the weather will be in 2 weeks or 4 weeks, it's getting more difficult, and it's a little similar with soil, because it's also such a complex system that changing one little thing can affect so many other different things that I absolutely understand that your answer is like this.
Yeah. At the moment, I would say that it's complex to give like you say, like the weather, no, because you are playing with two organisms needing different things at different times, but both are aiming to collaborate in the sense of symbiosis. So one develops, but because probably all the plants that you can think of, like maize, are deriving all the photosynthase into the production of maize itself, not to provide 20% of the carbohydrates to the fungi. And that's why we need to think in this direction, no? Like when we select for newest plant species, these plant species should be really pumps of photosynthase that not only derive to the fruit, but also to the fungi.
To the other.
And nowadays we are just searching the best ones, responding to, of course, less water, but even we could get this advantage for like for desertification, like to drought, to some abiotic stress. AMF are also capable of getting water from very small pores in the soil.
So so one more question about the establishment of the symbiosis. If one little let's see seedling is either inoculated or finds a spore, a mycorrhizal spore in the soil, how long does it take until there is a symbiosis? Is it possible to say that it takes a couple of weeks, or I don't know, or until there is a win-win situation, or
depends on very much again on the host plant and the fungal species, but it could happen within hours. So the establishment, the entering of the root could happen within hours. This is a process that is very well described. Once the fungi enters the root, there is a spike of calcium. There is a bunch of signaling inside, and the symbiosis starts to be operative within 2-3 days. And then if you want to have a full colonization of the roots, yes, you need to wait. Probably depends on the plant again, but 1 week could be. So I'm thinking on seedlings. I was working I was collaborating with a seedling company. They were just producing small explants for tomato, for watermelon. And we were applying the AMF directly in the seedling. They were putting the seeds, we were mixing with the substrate, and within 3 weeks, we were able to find the full colonization of the small root ball. So, and it was very nice distributed. So I guess 1 week, it was already inoculated, and then within the next 3 weeks, it was perfectly mycorrhized. So it's rather fast.
Okay.
Development of the hyphae in the soil, this takes a little bit more time, but inside the plant, it's really fast.
But can you then also say that as soon as this is established, that there are the positive effects of the symbiosis, or is it only that it's biologically established, but the hives are not, like as far as I understand, the hives need to go to some pores where there is a bacteria, where there is soluble peel. Do you know what I mean? Is there, because that's always what the farmer at the end of the day interests is how long does he take until, if he has a very good AMF basis in the soil, or if he has an inoculation, whatever, how long does it take that the corn, let's say, gets a positive effect from an AMF?
Here, for instance, depends very much also on the density of the plantation. I've seen here maize. It's really planted like, I don't know how much is the separation, 30 centimeters. So plants are really close. This is, I would say, it's the perfect scenario for mycorrhization, because you have a bunch of roots just pumping photosynthetics to the soil, taking up water, nutrients, and everything. And this is the perfect, I would say, because you have also water is very well conserved, because the area is fully covered of plants, and this may take hours or days, but again, depends again on the plant. And if you're applying this inocula in the form of Rutella or any other inoculant, again, depends very much on the amount of spores and propagules that you are putting in the soil. But when it becomes, because something that always, or it's been reported many times, is that at the very beginning of the symbiosis, even when you see the plant fully mycorrhized, you may see a growth depression. So plants are simply stopping a little bit the growth, and it takes probably 5-6 1 week days since the plants somehow establish each other with the symbiosis, and then it could happen that there is a depression in growth because some of these photosynthetics are invested into the fungi, and the fungi needs to develop. And this development of the fungal hyphae and the mycelial networks requires energy, which are of course obtained from the plant. Therefore, you may see at the very beginning a growth depression, but this is immediately depends again on the plants. Vanished and normally goes to normal growth if the conditions are optimal. And you will ask me, which are the optimal conditions? Depends on this.
Okay. One more question from my side. What you mentioned, what I find very amazing, because it shows that science of soil is so much on the beginning, is that researchers found out that it's not sugar basically that is traded or transferred to the mycorrhiza, but it's lipids. Is there any practical or any I don't know, is there any theoretical or practical implication of this knowledge, this new knowledge? Is there any big change now that to understand, or something that you now differently understand about fungus?
In 2018 or 17, someone used his new knowledge to I think it's a Japanese researcher. I can search for the paper. Since you can buy these lipids, you can buy, because the lipids the plant produces, they can be bought. It's these fatty acids, and he tried to grow the fungi without the plant just by providing the lipids, and they grew very well. And they could develop further. They could grow without the plant. So this was a way to test this mechanism, no? Like the lipids located by the plant to the fungi are the currency the fungi uses to grow, no? The source of energy that you're using to grow. And actually, they were publishing it, and I don't know, I haven't seen much after that, but it's a way to prove that it works. And this is one of the, and of course it's great because for researchers, we can use this information, at least I use it in my PhD, because there are then biomarkers. So you can extract lipids from the roots, which are only produced when the plants are mycorrhized. This is another way to look at this. You can also label the plants with certain C and see whether these labeled lipids are entering the soil. And you can then see whether the CO2 respired by the microbiota is coming from this certain C, which is in the lipids in the roots, is in the lipids in the soil, and this is bred in the. So yeah, there is a lot of research on this, and it's a quite active topic at the moment also because to decipher the metabolic, how they use these lipids to run all the metabolic pathways within the fungi. I cannot hear you.
Sorry, it is very interesting for me because I read a couple of papers and I saw presentations, and that's very new knowledge for me because everybody spoke about it's a trade of sugar or some kind of carbohydrates, but not lipids. It's very interesting.
Lipids are carbohydrates are taking place mostly in ectomycorrhiza. There you have mostly translocation of carbohydrates, trehalose and these kind of carbohydrates are. But in AMF are mostly the lipids.
Another question is from Clemens: is AMF only making phosphorus available for the plants or other nutrients as well?
Yeah, other nutrients as well. I mean, basically, phosphorus is the main one because it's really, really scarce in soils. And that's the way when plants and fungi co-evolved, but they are also important for nitrogen. And especially in those plants that hold the two different symbiosis, you can have nodules to fix nitrogen. And there are these double mycorrhization in some legumes where you may have AMF and also nodules that fix nitrogen. There are a lot of studies that say that there are super good synergies. So this could be one thing. Sorry, and the question.
If there are, coming.
And also there are, I think, copper, zinc. I was now doing one experiment and we found that they can also mobilize a lot of potassium from primary minerals. We will be publishing this probably next year. Also, they can mobilize potassium, but they are important for nitrogen also. They are quite important for nitrogen too, because they can mobilize. There's studies from Angela Hodges that they can access some organic matter and translocate nitrogen from organic matter, even if they don't have a super developed saprophytic machinery in terms of enzymes that can degrade the organic matter. They can mobilize some nitrogen from the organic.
Okay. But isn't there also often a cooperation between the AMF and bacteria in the soil, so that it's actually a symbiosis of three organisms: bacteria, fungi, plant?
I mean, the spores themselves, they have now there are studies on endosymbionts inside the spores. So there are now this is becoming more and more complex every day you open and read a new paper, because you have bacteria or endobacteria living within the spores, also bacteria colonizing the surface of the spores, and once the spore germinates, these bacteria are coming with the hyphae, and these are populations of bacteria which are specifically related to these fungi, and they provide some functions like the production of phosphatases, because one of the enzymes that AMF do not produce, or that they do it in a very low amounts, are phosphomonoesterases. These enzymes mineralize organic phosphorus. So any organic phosphorus that they can access, it's coming from this co-symbiosis with bacteria which are not normally free-living bacteria, but are bacteria closely related to the hyphae of the fungi itself. Yeah. So it's really complex, the interrelationships you may find, and you have a lot of groups of bacteria really attached to the hyphae and providing, because they feed from the exudates of the hyphae. The hyphae is exudating also some carbohydrates, some to the outside, and this is creating some kind of hotspot that promotes the growth of new bacteria, providing new functions to the symbiosis.
Then let's come to the last question, if there's nothing else from the others. It's about clomalin. You spoke about clomalin, that it's one of these carbohydrates which is very important for the soil structure, and it's very often mentioned in connection with mycorrhiza. But I also read that there's also some changes in research about these effects. Can you tell us something about that?
Yeah. Actually, glomalin, if you go and try to search the chemical structure of glomalin, you may find problems to find it, because it's like it's a box where we just call glomalin to any protein you may be able to extract from soils which are colonized by arbuscular mycorrhizal fungi. So I've read about the chaperone HP60 something, so it's like a heat-resistant protein. But yeah, in general, the functions of glomalin are well described. So they seem to provide some kind of gluing function, that they promote the adherence, the particles of the soil that where the hyphae were growing. They are somehow glued when this glomalin is secreted. And depends also very much on the fungal species, like Rhizophagos irregularis probably doesn't produce much, but then you have a species like Gigaspora margarita, this is a very nice name, and this is a beast producing glomalin, but probably it's not as good as mobilizing phosphorus as Rhizophagos irregularis. That's why it's so important, like to have mixtures of fungi when you inoculate the soil, that you don't have one. So these products, I've seen many products. We have patented this organism. It's the best one and works better. I don't believe this, because there's not one single organism doing everything. The mixture, the diversity of microbes, I think is the real key to respond to any changing environment. And with this, I would say you can be on the safe side. I don't know, I think Rutella or any other inoculant is made of 3-4 species, and they are tested. I would say it's always the best, because you have always the best of each species that can always respond, or you have an overlap of functions like production of more or less glomalin, more or less development of the mycelium. And as I said, it's also so dependent on the plant species, and this is not really well researched. So the research is not really well developed.
So I absolutely agree with you what in was concerning diversity, but the problem I see is that it's the more complex speaking about the microorganism product is, the worse
the response is more
you can so that there's always you need to find one way. Either you go in one direction with one or two or three organisms and you study them very deeply, and you can find effects and you can understand what happens why. And the problem is, we worked, for example, last year with a product with 700 different species, and it's from the States, and it's basically they extracted from different soils in all the organisms and try to put it into a product under standardized conditions. But it sounds very interesting, but it's very hard to predict anything about it. I think because there is so many complex interactions between them, the organism each other, that so it's very, very difficult.
Look, I think the best inocula you can produce is the one you have in your field, because you have a natural abundance and a natural presence of organisms like Arbuscular mycorrhiza. And there are several ways to reproduce the microbiota you have in a certain place nearby your farm. And this one is already adapted to those conditions. And in an ideal world, every farmer should have a small portion of their land reserved to reproduce, to enrich their natural microbiota using these trap plant systems or any system to just enrich it and then spread it all over their fields. This could be, in an ideal world, the best way, because you don't really need to, you're sure you're putting everything that was already there, and you just put it in excess. And I think, from research studies, this is the way. Normally, you end up finding the best results, because you not only increase the diversity of any inoculant you can buy or anything, you are using the organisms already adapted to those conditions. And because it happens many times, is that any inoculant you can add, not always, but it could happen, it's displaced by the natural microbiota you have in your soil. Of course, if your soil it's been used for an intensive agriculture over years, probably the amount of propagules you have for AMF is so low that anything you can bring inside is the best thing you can put. So of course, I'm prone, I'm fan, and I'm supporting of any kind of inoculant, of course. But then, try to introduce as much biodiversity as possible would be one thing, and of course the one which is already adapted to the region.
Absolutely. The thing is, we also have to get a little bigger view that soil is not only EMF. It's huge more, and we have to try to find a system, agricultural system, where we get all the biology and the diverse biology done. The problem is that in agriculture we are at the very, very beginning, very beginning, to get in to start a system that improves the biology and all the biology, because I think that's important knowledge. That most important is that if you have a balanced, diverse biology, you get a redundant soil, and at the end of the day, it's economically also the best.
It will respond more resiliently to any disturbance. But also it's about the mixture of crops, or like Norman is doing with catch crops, in combination with AMF, combining with fallow. Everything just brings more and more benefits. So I think we are in the very good direction. nowadays we have also the tools to describe and to define and to understand these complex relationships. Even though we cannot fully understand them, we now have the tools to start to understand them, and that's why it's a very challenging and interesting moment. Also, I really feel there is still a lot to do, and I think as soon as there are some parameters that we could keep under control, I think big companies, the seed companies, are the ones who probably would need to be convinced, because if they are convinced, then to select, to breed for plant species which are adapted to coexist with any kind of symbiont, then I think we would be in the right direction. Yeah,
that's a good final for this webinar, but I'm afraid that most of the seed companies do also sell fertilizers and plant protection products, so that they are not really interested in this kind of system, I guess. But I think society and the common sense is also very much changing in this direction. It's it's also something that I and we feel a lot with the farmers, that they are interested in making their soil better, in that they're interested in understanding the soil, because that's new knowledge, that soil itself is something, is a living organism, and we can use it in order to get better yields at the end of the day. So there is a lot, at least what I feel is that there's a huge change in the society and also around the farmers to this topic.
Yeah, of course. Yeah, there is, and the good thing is that an ongoing process. So I think it's not going to stop, and it will grow, but probably not at the speed we would like to. But I think it's an ongoing process that it's not going to stop, because we need alternatives to either fertilizers or management solutions that make our soil sustainable over time.
Yeah. So thank you very much, Alberto. It was very exciting.
Thank you for your time also, and thank you.
Thank you. I hope all the others found it interesting as well. As I said, we will put it on YouTube the next week. Probably in German, but maybe also in English, because we had a lot of conversation. I don't know yet. Anyway, I wish you a nice evening, everybody, and thank you for being with us and learning a lot with Alberto. Thank you very much.
Thank you to all of you, and have a nice evening. Bye bye.
Bye
bye, Christoph. Thank you.
Thank you very much for listening and watching this video from Alberto and me. I hope you enjoyed it. I hope you could get some information for you. You could learn something, and maybe you will seek another or watch another video from Farm to Farm. Mostly they are in German, but anyway, I hope to see you soon.